This week for case study 4, I'm going to be discussing penicillin and how it works to kill bacteria in the body. So first, penicillin belongs to the beta-lactam antibiotic class. And this specific antibiotic class works by indirectly bursting the cell wall of a bacteria. It acts directly on the peptidoglycans, which are essential for the structure of a bacteria cell. So a little more about the peptidoglycan in a bacteria. So peptidoglycans create a mesh-like structure around the plasma membrane of a bacteria's cell. They increase the strength of the cell wall and prevent external fluids and particles from entering the cell. And specifically for gram-positive bacteria, they contain a thick cell wall and a high level of peptidoglycan. In contrast, gram-negative bacteria contain a thin cell wall and low levels of peptidoglycan. And so when bacteria goes through cell division, they multiply, and newly created peptidoglycans reconstruct the cell wall. So penicillin contains a 4-membered beta-lactam ring, and it can kill bacteria by binding the beta-lactam ring to DD transpeptidase. And that is an enzyme that inhibits the cross-linking of a peptidoglycan in a cell wall. It prevents a new cell wall from forming. So as the cell wall begins to break down, osmotic pressure pushes water into the cell and it kills the cell. So penicillin can also, bacteria can create resistance towards penicillin. A way that they can do this is through the rapid reproduction of bacteria mutations that are present in the cell, and genetic mutations occur when antibiotics are in use. So because antibiotics are present, that creates a survival advantage for the bacteria, creating a resistant strain. So the resistant strain can only be killed through an alternative of an antibiotic or a stronger antibiotic other than penicillin. Antibiotic resistance increases when the antibiotic use is repeated or it's misused. Ways a bacteria can develop immunity. Penicillinase is one way, and that's when bacteria can produce an enzyme that breaks down the penicillin. It spreads through a small ring of DNA called conjugation. Altered bacteria structure, so changes that a penicillin protein undergo in their peptidoglycan wall can create immunity within the bacteria. So penicillin can no longer bind to the peptidoglycan wall because something happened with the structure. Penicillin removal, which is where bacteria can use the efflux pumps and they release a substance that can dispose penicillin. And then more specifically, gram-negative bacteria. Gram-negative bacteria contains an outer membrane. So many antibiotics pass the outer membrane to access their target. So, for example, hydrophilic antibiotics, such as the beta-lactam, pass through porins. So when there's changes in the outer membrane, like mutations of the porin or changes to the hydrophobic properties, this is when resistance can occur as well. And then using the Chimera There it is. Here is the penicillin structure. And then to end with my references.